has gone.
Yes, we can hear you.
We're looking at the phage harmful properties that we have to consider: presence of those virulence factors, the ability to transfer genes, especially if there are those undesirable genes, which can be transferred to bacterial strains and therefore create new pathogenic strains or emerging strains. And then having these new genes into the bacterial gene pool, which may be a positive or a negative outcome or a neutral outcome. So our worry and why we are pushing for bacteriophages is to fight antimicrobial resistance. So it would be a major scandal if we are using bacteriophages that are carrying antimicrobial genes. Then when we look at the persistence and survival in the environment, this can depend on the nature of the bacteriophages and the conditions in the ecosystem. Very often, as we are carrying out our research, we are looking at the settings or the physical chemical conditions in the settings where we plan to utilize these bacteriophages. We look at the pH. Are they able to survive various pH, especially the ones that are found in that setting, the ionic strength, temperature stability, the soil structure, because at times these bacteriophages may adsorb to certain types of soils, and especially if they adsorb, then how are we able probably to eliminate them in case we want to get rid of them? Then we also look at their adsorption properties, and if there is exposure to sunlight or the intensity that they can withstand. So maybe here I remember on the first day for agricultural applications, we thought, I think there were questions about their stability in the sun if we apply them on the crops. So that means we have to look into all these factors and select bacteriophages accordingly, or probably utilize their inability to survive under these settings to get rid of them. So very often there is escape inactivation. I think that's why we would think in sewage, probably the conditions may not be very favorable, but that is a very rich source of the bacteriophages. And they can also survive water treatment processes, food purification processes, pasteurization, and worse still, if we use manure as fertilizers for the vegetables, we can introduce those phages with undesirable characteristics. Then dissemination in the environment, from the lab, and then into the where we need to apply them. We are really looking at phages with narrow host range to such that we minimize on attack on non-target organisms. However, in some circumstances, there is need for a phage that has a wide host range. And I would think so, like in agricultural production systems, where we may have different strains. We may not have myriads or millions of phage candidates to attack the different bacteriophages, the different pathogens. So we may need some that have a little bit a wide host range to serve the purpose. There are also phages that have been developed by genetic modification, and either to widen the host range, but also for other purposes, like maybe gene transformation and transduction processes. We are also aware that phages are not limited to a specific habitat. So if they are used in terrestrial environments, they will end up into the marine or the aquatic systems. So how are we going to take care of that? That also has to be at the back of our minds, especially where we are looking at application in the agricultural settings. How are we going to ensure that we are limiting this flow into different ecosystems? The problem with the broad host range phages, especially if they have capacity to transfer genes, they will, in as much as they will promote genetic diversity, which may be desirable in some instances, but in the case they are virulent genes, then we have a problem. So the hospital settings, whatever is the influence from the hospital, and within the hospital, we are mainly looking at the pathogens that would be a risk if these phages picked up the virulent genes from the pathogens. Then genetic modification has come in. As I mentioned earlier, at times there is need to widen the host range, and genetic modifications or coming up with the artificial bacteriophages has been mentioned. Then we may increase the biological risks because of probably introducing new genes or new properties, some of which are undesirable. And then the type of activity, where are we using them? Is it on the farm? Is it in the hospital? Is it just a lab? Is it within research, the research lab? All these we have to put into consideration because we know that phages have been used in epidemiological studies to detect spread of pathogens or the exposure of individuals, especially like in cases of food poisoning. But the laboratory practices and tools that we use, we should follow the good manufacturing practices. We should avoid accidental injuries. You may introduce pathogen, phages into your body, which are not yet characterized and you are not sure they are safe. There's methods of pipetting, vortexing, centrifugation, physical containment. All those ones have to be observed in order for us to avoid dissemination and contamination of the environment. But we are sure that in as much the phages are safe, but the bacteria that we may be handling may be at different biosafety levels and may need different laboratory settings. So depending on the pathogen that one is handling, make sure that you are using the required or recommended biosafety lab, the lab biosafety levels in order to contain bacterial contamination as well as the spread of the bacteriophages. As it has stored here. So there are some biosafety recommendations and containment measures. We need to characterize the phage genomes in order to know the toxins and the virulence genes. Some of these may be of high risk, others may be of low risk. So the need for the personal protective equipment is important. Then we have the use of lytic phages versus temperate phages. Janet presented about the Clostridium difficile bacteriophage, that they were mainly template, but they had to manipulate them to be of use. So that means they removed that capacity of gene to transfer genes in order to utilize those particular bacteriophages. Then narrow host range bacteriophages are considered to be more safer and there is limited risk of dissemination unless their hosts exist. So that's why there is need to use native phages, or if necessary, generate modified phages, but which will not be able to attack as very many different bacteria as possible. Then we have to look into those biological risks, comparing the containment level, which I've already mentioned. We look at what is required, especially in relation to the biology of the bacteria. And we shouldn't forget the good laboratory practices. We should have trained personnel, and the safety equipment, facility design specifications should also be looked into in order for us to contain these biorisks. And after each activity, we need to inactivate all biological wastes. So if we've applied on a farm, let me think of the aqua farm in a pond. So we must have a way of decontaminating the waters before we release it to the environment. So that means when we are looking into application of these phages, we must think through how best we can utilize them to in order not to inconvenience the decontamination process, because having a pond that is probably harboring 2,000 fish, that is big enough. I wonder how one can decontaminate that water. Then the national, for the regulatory frameworks, as I said, not much exists as far as bacteriophages are concerned. And when I tried to read around, I realized that the Uganda National Drug Authority, they are looking into these conventional drugs, but they have also introduced regulations for herbal medicines. And this is, for the herbal medicines, it's quite recent. Not many have taken them up, and it would be difficult to, I think, I wonder where we can now fit the bacteriophages. Should we push them into herbal medicines? They do not fit in there, yet they do not fit into the conventional medicines. So, but as for research, I think the National Council of Science and Technology gives guidelines on for research, although they concentrate more on the humans, and we realize that phage application in humans may not be as near as it is for agricultural applications. So we have to think through what we need to do in order to fit within our fit within our settings. And I think here the Ministry of Health, the Ministry of Agriculture, they have to work hand in hand in order, together with us, I think the ones who are spearheading phage research, to come up with the regulations that will guide the research as well as applications. And we have fragmented policies, and these policies, probably as we are trying to come up with the regulations, they have where we fit or we may fit, we have to bring them up. But most importantly, like Dr. Atunga said from when Dr. Toby presented, we need to sit together with them to see how we can come up with the guidelines and regulations for bacteriophage research and utilization in our settings. That is all I managed to come up with. I thank the symposium organizers and the mentors. I think Phages for Global Health, Dr. Betty Kuta, Professor Martha, they really did a good work in us, and we are trying to not disappoint them, to push it further. So I thank you for your patience and perseverance, because the technology had completely failed us. And I cannot forget my Phage Team Uganda members who are always supportive when I'm given us an assignment to do. Thank you very much. Hello.
Yes, Doctor.
Yeah, so I've ended my presentation. Thank you.
Okay. Thank you, Dr. Jesca, for your very elaborate presentation. We just, on my end, we just have one question from the chat. It says, from Lillian Musila, how do we monitor for detrimental effects of phages in animals and humans? For example, immune modulation, integrative events, and have there been any studies looking at the very long-term effects of phage treatment?
I think phage applications have just, I cannot say that it is long-term, and the applications are just coming up, but many of them, because of the reasons as to why applications have not come up, I think it is the regulatory related issues. Because especially for human applications and maybe animal applications, you need to do those clinical trials to see that your phage has really the effects, and the pharmacokinetics of the bacteriophages, the pharmacokinetics of bacteriophages is quite different. Is quite different from what we see with the conventional drugs. So the dosages, monitoring how it reduces within the bodies, and that one I have not really come across a comprehensive review. That is the one that would help us. But I think with the time, more data is coming up, and maybe in 3, 4, 5 years, we should be having information on the long-term effects. But I think, like for environmental contamination, the principle was that as long as the host bacteria is removed or is handled by the bacteriophages, then the bacteriophages will also get out of action. So that's where probably we need to look at the environmental impacts, the microbiome changes as we apply these bacteriophages. Lillian, have I attempted your question?
Yeah, thank you so much. In fact, I was actually just wondering if anybody has come up. I think I agree that the treatments are still in infancy. I know that where phage treatment has been happening for long, like in the former Soviet Union countries, I don't know if there's any data from there that anybody has come across.
It could be in Russian. But at least somewhere, I think with the first ever green meeting that I attended, there were some scientists from the Eriava Institute who presented on a kid who had, I think, was it an opening in the throat, a wound that was not healing on the throat. and they tried to treat that kid, and they realized, okay, antibodies were detected, but they did not affect the activity of the phages on healing that particular wound. Maybe superficial, since they started more with the superficial wounds, which may not, whereby the immune response may not be so much influenced if we apply topically. That could be the reason we do not have a lot of literature, but we need to scratch further, because what the previous speaker presented, I think, gives us an insight on what is happening within the immune system in our bodies.
Thank you, Prof.
Okay, I think from the chat, I see Joseph Michael commenting that most documents from the Eastern Bloc on phage work are either in Russian or German. And I see Geoffrey Otim from Zimbabwe, Africa, saying, is there any initiative within the continent developing phage libraries?
I think the efforts, whoever is carrying out research on phages is trying to have a repository, not a standard one, but at least we have a collection of bacteriophages. And like Toby presented earlier,
so Dr.
we are looking into...
However, I think I'd just like to comment, Dr. Just...
Yes, please. Come on.
You are breaking a little bit. You are breaking a little bit. So we have you back.
Okay, so I was saying that whoever is working on bacteriophages is trying to keep a culture collection or phage collection in our own local settings. But like Toby presented earlier, we are looking into having repositories well organized and probably which can feed into a continental or regional banks for bacteriophages. So it is okay. Someone is asking about advanced phage scientists with opportunities for lab attachment, where we can send out to acquire skills in phage research. This could be yes and no, because especially in our region, we've just started. Unless a lab was already advanced, it has those particular facilities. Maybe we can also have those phage scientists, advanced phage scientists. I wouldn't count myself as an advanced phage scientist because there are so many areas in phage research that I'm not certain about or I'm not confident about, but we can easily learn. Maybe her name has skipped. Those at the Iriri in Kenya and maybe Kemri, those ones could do, and also Atunga's Institute, maybe those ones may have some of those facilities and where advanced phage research can be done. But otherwise, we are still on the basic isolation and characterization using the ordinary, if it was bacteria, the phenotypic methods.
Okay, I think we've lost you, Ivy, and since we are come to the end of today's talk, maybe we'll just move on to the next segment, and then we can quickly close because we are really way past the time. Professor Nakavuma, I want to thank you so much for this enlightening talk. I think the issue with maybe us in African Phage Forum, where we are either, still thinking of concepts or we are still in the earliest of the earliest phage research or mid-career. We don't even think about regulatory pathways. You know, all you want is to see what a phage looks like and maybe do a couple of, you know, basic characterization. I mean, that's what really excites us. I think I'm speaking for me, I don't know about the rest, but we don't really look at the bigger picture, which you have brought out. Personally, I didn't even, you know, each time, you know, you apply for a grant and they ask for the ethical and the safety checklist. For me, I always just tick the phages as, you know, safe for everything without even thinking, you know, about the details that you've given us. So for me, I really, and on behalf of very many of us, I want to thank you so much for broadening our mind to not just thinking about, you know, the in vitro experiments that we do, but to think wider into the regulatories, into the safety nets and all that. I want to really thank today's speakers, you know, Dr. Toby, Dr. Jonas, and Prof. Nakavuma, but also to thank those who have been with us since Monday, yesterday, and today. I'll repeat that. But also to thank the audience, the attendees, and to, you know, for sticking in there, you know, for all this time. Really, really want to thank you so much as members of African Phage Forum. It is an umbrella, and yes, I know there are coordinators who are behind all this, but we tend to make it tends to be an umbrella for all, you know. That's why you don't see particular faces come out. It belongs to us. It is us who are supposed to move, you know, the continent forward. So I really, really want to thank every one of you who has taken the time to just stand with African Phage Forum, to give your feedback. Definitely next year we want to get back to you with this feedback, and then we see how do we move forward? How do we move from novice to in vitro, to commercialization, to public sensitization, to regulatory pathways? Like, these are now the real conversations. These are now the real conversations. And we want to apologize for anything that has not gone right on the coordinator's side. But we know that for every first time, there will always be these technicalities, and, you know, coming from, you know, there'll always be these hiccups, but we want to appreciate that. I think, you know, on average, it's gone well. Maybe I need to now move on to the coordinators, and I think the coordinators are represented by Newton, who can now just give us a closing, just one final word. And